DNA is like a nachos box where it is made out of individual building blocks called nucleotides, similar to how a nachos box is made of individual nachos. Each nucleotide is further made up of a sugar, a phosphate group, and a nitrogenous base, similar to how a perfect nacho has an adequate amount of salsa and cheese in addition to the chip itself. The 2' carbon of a DNA nucleotide ribose sugar is deoxidized, hence the name deoxyribose nucleic acid. The 1' carbon of the deoxyribose sugar is where the sugar binds to a nitrogenous base. There are two groups of nitrogenous bases. Purines consist of adenine and guanine, and pyrimidines consist of cytosine and thymine in DNA. DNA is made up of two strands, which are said to be antiparallel, meaning that if DNA was to be unwinded to look like a ladder, where the deoxyribose sugar and the phosphate groups make up a rail, the rails run in opposite directions of each other. Base cycling, which is the cycling of the nitrogenous bases on top of each other, is a part of the DNA structure as well and allows for hydrophobic interactions to maximize. This is important as hydrophobic interactions are the main force in stabilizing the double helix structure of DNA. Bonding of nitrogenous bases of the two strands of DNA follows a set of base-pairing rules. In DNA, adenine and thymine bind together using 2 hydrogen bonds, and cytosine and guanine bind together using 3 hydrogen bonds. However, it is important to note that hydrogen bonds are relatively weak and are not the main force joining the two strands of DNA together. Two nucleotides on the same strand will bind using phosphodiester bonds, where the phosphate backbone binds the 3' carbon of the previous nucleotide to the 5' carbon of the new nucleotide. Therefore, DNA is read and written in the 5' to 3' direction. As the double helix structure of DNA grows in the 5' to 3' direction, the different parts of the helix are not uniform in distance, and there are major and minor grooves in the structure of DNA. The helical shape of DNA also allows for DNA to have direction and be either left-handed or right-handed. If DNA was to be like a Twizzlers candy, if the candy was rotating counterclockwise while twisting and moving up, and there were lines pointing from left to right, the DNA would be right-handed, while otherwise it would be left-handed. Left-handed DNA is very rare. DNA is negatively charged due to the phosphate backbone. Usually in the cells, a high cell concentration buffer stabilizes the DNA structure by binding to and shielding the negative charges, as otherwise the repulsion between the negative charges and the strands makes it more energetically favorable for the two strands to separate. Therefore, by shielding the negative charges, a higher cell concentration will increase the temperature that the DNA strands can tolerate before they denature. While if the cell concentration is low and the negative charges are not shielded, DNA can be denatured at a lower temperature, as it would already be unstable due to the negative charges.